السلام ورحمة الله وبركاته. أنا اسمي عبد الرحمن عيد. اليوم نتكلم عن محاضرة غلام live proteins. Okay, two major things we need to understand before we begin. The formation of the crystal, the structure of the cholesterol, and there are two types. We have hydroxyl group type, which is called the cholesterol, the normal cholesterol, and we have the ester group, and the ester group is like this. So it's like R O O C. You can see. So the ester group is, we call them as hydrophobic, they're not needed or getting in contact with water, and hydroxyl, they contact with water. And this is very important because we will use this information to exceeding forwardly with VLDL, chylomicron, and go on. Another thing is the isoproteins. Isoproteins, they divide to four major things. Actually, you don't need to remember really the numbers, but yeah, you need to understand what the function, then you need to remember. B, E, A, and C. Now, B and E, the first thing I do is that because they are the same function, I put them in the first, then A, then C. B and C, they are do the same function for one thing. They are activators of receptor on the liver. So, receptor, activator. They have the same function. And A is, just skip it, not now. And C, the activator of lipoprotein lipase enzyme, and this is in the adipose tissue. And we'll see the function. And A, now A is a little bit different. A is have in the HDL surface, and the reason why we'll use them in the IDL formation to LDL. And we'll see how they actually do. Now, the first thing we see here is the lymphatic system, because we use the small intestine, eventually go into the lymphatic system and then circulation. And this is the chylomicron. The first thing we need to do is the chylomicron need to understand that it is contain highly amount of triacylglycerol. Highly amount of triacylglycerol. Okay? So chylomicron, this one, contain large amount of triacylglycerol and also cholesterol. Now, how they actually go to our system? They go by simply the absorption. When you eat a high or fatty meal, and the normal fatty meal is 250, is actually milligram. It's actually not that much. You actually eat one hamburger and eventually just exceeding 4. Yeah, it's just normal. But when you eat a high fatty meal, the thing is going to use is that the triglycerol will be break down, and then the glycerol will be absorbed, the fat will be absorbed, then reform. This reformation to form something called chylomicron. The chylomicron, the first thing part, it will go to the lymphatic system. But before we go to the lymphatic system, it gives a specific protein called lipoprotein B. Now, this is very important. There was two types, 48 and 100. 48 for the small intestine. Need to remember that. 48 is small intestine because it's high yield. We will use it eventually. Now, when we go to the lymphatic system, it now go to the blood. Before going to the blood, during the lymphatic system, we'll see specific lipoproteins, and this is called our lipoprotein called HDL. Now, what does HDL contain? HDL contains two lipoproteins, E and C, specifically two. Now, what they do specific normal function. They pump with each other. When they are pumping with each other, they just simply, the chylomicron give a little bit of fat to the HDL, and the E and C2 will be taken by chylomicron during the lymphatic system. This is during all in lymphatic system, not the blood circulation, in lymphatic. After going to the lymphatic, when they do and pump with each other, take all the specific parts, then the chylomicron go to the blood. Where are they going? They go into the adipose tissue simply. This is the adipose tissue, and this is the chylomicron. And chylomicron is contained this specific, what is this thing? So LDL, lipoprotein, simply the enzyme lipoprotein lipase. Very important to know this. When we say diabetes have a specific function or maybe function to a specific disorder in lipoprotein lipase, why? Lipoprotein lipase can be activated by C2, also can be regulated by insulin. So you can imagine if the patient has diabetes, insulin can damage lipoprotein, and not also that, it can C2 would, well, because the patient has C2 damage and insulin damage in diabetes, eventually he will be severely contain the specific type, and which is called type 1 hyperlipidemia. Okay, now after they do that, after they go into the adipose tissue and triglyceride is taken, after they go to the adipose tissue and triacylglycerol is taken, they degrade it, and eventually free fats will be taken, and glycerol will be stored as glycerol phosphate. The major thing that it can be glycerol phosphate go to the glycolysis by gluconeogenesis, and eventually by gluconeogenesis, they will go to something called dihydroxyacetone. And that's a normal thing. They will want you to know that is you can take glycerol to form carbohydrates and something. Now, the chylomicron contain ApoC2 and ApoE and ApoP48, all of them at this point. Before they go into something called chylomicron remnant, they give the ApoC2 to the HDL. And the remain is ApoE, because we say that ApoE and ApoP48, both of them activate receptor on the liver, this one. And you can imagine if the ApoE is defected, the receptor will not form, right? So chylomicron remnant, eventually, they contain low amount of triacylglycerol and high amount of glycerol, because we say go into the position and store triacylglycerol. So they contain ApoE and ApoB48. Okay, so this is the whole story eventually. Now we'll go to the liver. What is happening in the liver? You see, the liver contains specific organelle, called lysosome. And what lysosome do? They eventually give cholesterol inside the liver, and the cholesterol be taken for the lysosome, degraded, and eventually be forming as, you can say, as bile. Okay, now simply, this is the whole picture, but you need to understand what is the function of cholesterol ester now. Cholesterol ester is give low fluidity. What does that mean? Let's imagine this is membrane, plasma membrane. What does plasma membrane do? Plasma contain protein and fat. And we remember in the foundation that cholesterol gives much more flexibility and fluidity to the plasma membrane. So why we do ester? Because ester is hydrophobic. So it didn't give any kind of fluidity to the plasma membrane. The one that gives fluidity is HO, which is simply the hydroxyl group. Ester is hydrophobic. That's why we need that. Imagine this. With the much more fluidity, the cell will take much more water, the swelling of the cell, then the rupture of the cell. Necrosis will happen. So everything will be homeostasis. So this is simply the chylomicron pathway. Now we go to a specific pathway now, which is the VLDL, and eventually we'll be discussing all the damaging disorders by one at the end. I'll tell you what is defected in your dual. VLDL, and we will compare VLDL with chylomicron because they have a little bit similarities and a little bit different things. VLDL is from the liver, is originated, and eventually they contain a specific apoprotein called apoprotein 100. You need to remember this is high yield. Apoprotein 100 is multi in VLDL. Okay, multi, not single unit, multi, and that is due to pathology. We'll use that. Okay, VLDL contain apoprotein 100, going from the liver, eventually going to the bloodstream, not the lymphatic, immediately to the bloodstream, and you see specific specific lipoprotein called HDL, high density lipoprotein. And what does they contain? The same thing. They contain ApoC2 and ApoE. What they do, they pump with each other. They're just giving, one of them giving fats, and the other them take ApoE and ApoC, and what they do with it, which is the VLDL, they go to this specific enzyme. in adipose tissue. The same thing happened. Triglycerides being degraded, and eventually fat being stored in the adipose tissue, and the glycerol be stored, or by gluconeogenesis to form carbohydrates. Then eventually form IDL. Before they do that, they see HDL also, and they give HDL ApoC2. And what ADL contain? ApoE and 100, which is B. ApoE, ApoB 100. Here is the most important thing you need to know. IDL contain cholesterol. Now, here the difference. This is HDL. HDL contain on its surface a specific apoprotein called apoprotein A. This specific apoprotein is so important. Why? Because you need to imagine, if IDL contain cholesterol, we said it before, if cholesterol is high in the liver or any kind of tissue, it will give fluidity, right? Then the necrosis will happen, and eventually all this pathway. We need to form cholesterol to cholesterol ester. We didn't want fluidity by the step of fluidity. This is just not make sense. So HDL contain apoprotein A. Now, what is the function of apoprotein A? Will activate a specific enzyme called LCAT. You just remember L and CAT, just easy. And what is the difference between LCAT and ACAT? LCAT is outside. This means not intracellularly, inside the organ. No, just in the bloodstream and go on. So HDL contains the LCAT enzyme. IDL can go to two specific pathways. Either can form an LDL or go to the liver and be forming as a bile and just secreted out. Now, apoprotein A will retain cholesterol. How they do that? Just so beautiful way how they do that. Simply IDL and in the bloodstream they do it. They just pump with each other. They give cholesterol and eventually give cholesterol in the HDL. The HDL take it with apoprotein A. The apoprotein A going activate LCAT. The cholesterol will be formed in the esterification mechanism. Hydroxide will be taken, giving ester and cholesterol ester. And then eventually pump with LDL, IDL eventually and giving cholesterol again as ester cholesterol, or cholesterol ester. So they do this specific mechanism. You take it. No, you take it. Hey, you take it. And they do that. Always they pump each other inside the blood until they go to the liver or do another formation. Now IDL is formed and they contain, I already told you, it's very important, they contain ApoE and ApoB 100. It is so important. When the ApoE is being taken off from the IDL, will form something called LDL. LDL contains a single unit with ApoB 100. Single unit with ApoB 100. This is very important here. So it will form the specific pathway by E taken off, and LDL will be formed only LDL contain cholesterol, ester, and ApoB 100. A single unit because due to this specific pathology. Okay. Now we understand most of the things. Now, either IDL form LDL or going to the liver. And how they go to the liver, I already told you. ApoE and ApoB 100 is activated on the receptor. They just bind to the receptor, and then eventually they form the specific pathway that, hey, lysosome, and eventually form as a bile secretion out. So, just simple things. The problem is LDL. Let's take a specific formation of LDL and what is called oxidized LDL, because this is high yield and associated with the pathology. Okay, LDL either can be a peripheral cells, as you can see, going to a degradation mechanism or forming oxidized LDL. Let's just take the oxidized LDL because it's simple due to the overall pathology. I already told you ApoB 100 is single unit, right? So you can imagine what is the function of ApoB? Is activated receptor, right? So imagine if ApoB is oxidized. What is being oxidized? There are specific cells called oxidizing cell. You can say that most of the free radicals, they take it and they just take electrons, and eventually they give electrons to just decrease the amount of free radicals in our body. Sometimes they see LDL and they want to take a little bit of electrons from the ApoB 100. The problem is when they do that, they can oxidize the specific apoprotein 100. Because it's single unit, if the ApoB100 is single unit and oxidized, the receptor will not recognize the specific protein. They will see it as a foreign body. It's a thing that is simply outside our body. What they do, well, the macrophage will come, and if the macrophage will come, oxidized LDL will see them. They say, okay, what is this thing? It's not our body, it's just bullshit. They will take it out and eat it. And they will eat it, and they'll eat it, and they'll eat it. Eventually, the more they eat it, the exceeding expanding of the LDL oxidized one inside the macrophage. And TL or T helper cell will come and activate it, and eventually more accumulated of macrophages and eventually will increase. In the endothelial cell, the more accumulation, the more damage to the endothelial cell, and the more damage because the necrosis of the macrophage to form as a foam cell by increasing of eating its specific pathway. And necrosis, and what the necrosis will do, all the oxidative LDL will come outside of the specific or endothelial wall, and the accumulation eventually exceed and exceed and expand and expand. The more macrophage will come, and the secretion out of all the specific enzymes will be secreting out from the macrophage, which is called, well, we can say as enzyme that is bad for us, hydrolyzing enzyme, will be expanding and expanding and expanding until we form something called atherosclerosis. So this is the pathway simply, as you can see in this specific part here. Now LDL. In LDL, in receptor, we have a receptor. In this specific receptor, in this receptor is we have as the LDL. The receptor have percentage. The more the LDL has been taken as recycling mechanism by ApoB100 activator for this specific receptor will be taken, and eventually the more that it's recycling, the more losing percentage by this specific receptor by recycling inside the cells. Okay, just imagine why they are taking LDL, why they want that. Because the LDL contains fats, and we want that because formation of ATP. But also we don't want that much of fluidity in the membrane. We want ester bond. So they take LDL and they take it from the extracellular matrix. We don't want to form it inside our body or inside our cells because it's too expensive to form it. So it is easy to just eat a high fatty meal and eventually eat it from the specific part. Then the LDL either can be stored or can be degraded and formed as a bio. The problem is if the LDL receptor is defective by always the recycling, lose a percentage of the specific receptor, eventually will form a damage to the specific receptor. A receptor will be not actually as a gate, and the gate is just broken. You cannot recognize the ApoB100. You know what's happening? It is so beautiful. The outside of the cell will accumulate LDL, and the inside of the cell will be so starving of the LDL. They start to form fatty tissue, fatty mechanism fatty acids inside the cell, and even the LDL is so much increasing outside. They form cholesterol inside because they cannot take it. So now the cell is accumulated with cholesterol, and also outside is accumulated with glycerol. It just double things. And we form something called type 2 hypersensitivity, which is type 2, you can say, is hypercholesterolemia. This is because of receptor damage. So most of the things in the pathway just going like this specific part. So I think this is sufficient for most of the things. Okay, thank you. If there's something I left it, please tell me, and eventually I will simply as giving the information for it.